Yeah, perfect. Indeed, as Katarina said already, I want to talk about field phenotyping photosynthesis. And then what I would like to do with you today is to get from this process that you see on the left side, you see a plant with some blue light, and the photosynthesis is happening. It is actually in the green parts. With the blue light, we can get a photosynthesis signal out of it. I will talk a bit about it more that you understand better what I mean with that. And I will bring this to a field where I show you how we brought it to the field, the middle picture. And on the right side, you actually see a phenorob field, one of the phenorob lectures, and we have been measuring in this phenorob field. I will not present too many examples of the phenorob field, but I would like to say already now that I would be very happy to see in the field this year, because we will have a phenorob central experiment again this year. That is background. Let me start with photosynthesis. Why photosynthesis? Well, you know this textbook image, I think, that photosynthesis is this process where CO2 is captured by the leaves, oxygen is produced, and sugars. This is a process where you really have gas exchanges, and this is how it's typical measured and also typical referred to. Below this textbook image, you see the other part where light is absorbed by the leaf, really the energy source to get this sugar transversion. If the light is absorbed, we have one phenomenon. You also get a fluorescent signal. And this fluorescent signal has the great advantage that you can, from a large distance to remote sensing, to very large scales, even to the extreme scale of satellite. In 2026, actually, among others, by the contribution of Urashi, there will be a satellite launched that will be measuring specifically fluorescence, this interesting photosynthetic efficiency trait. I will not talk about that today because we are talking about phenotyping, and the scale of the satellite is about 300 by 300 meters. So what I would like to do is to go back to that gas exchange, that little process where you see here a picture of the early times when I started in Jülich about 10 years ago. You have a cuvette and you put a leaf inside there, and then you can measure gas exchange processes. Rather laborious and also time consuming. So I directly compared it with a device that you see in the back that Katarina mentioned already, a light-induced fluorescent transient device, a lift. And we did here the first measurements, we compared it. Can you measure gas exchange? Can you compare that fluorescence? And these are the results of these early measurements, where on the y-axis is gas exchange and on the x-axis is fluorescence. And you see that they are highly correlated, not surprising, under many different conditions, on the different light and different temperature conditions. So the short take-home message of this, if we measure fluorescence, we get a very good idea about the photosynthesis processes in the plant. And this we can use in field phenotyping. And what do we actually mean by field phenotyping? This means if I can say we can use it, it says we would like to, in phenotyping, identify different genotypes on the different environmental conditions. If I split it down a little bit further, that will mean a trait, in this case photosynthesis. And in this talk that I will focus on actually mostly one sensor, the lift. Of course, there may be more other in field phenotyping context in general, there may be more sensors or may be more traits that we can quantify. But as I said, today it will be about photosynthesis only. I will combine it with different positioning systems that you will see different examples of. And I thought it would be nice in this talk to give you examples of experiments we've done and where we have used the lift. Examples with some illustrations, how it looked like. Some published data, some unpublished data that go from model data to heat stress, drought stress, disease. So the first part is abiotic stresses. Then we have an example of biotic stresses, and I will end with a response to CO2 and in relation to how photosynthesis responds. I will do this mainly focusing on this abiotic or biotic stresses, but these are all environmental factors that we modified experimentally. And that's also one part that you should not ignore in phenotyping. Phenotyping is genotype and environment response. And here you will see that I sometimes have forgotten that also, but in the total concept of field phenotyping, I want to emphasize, look at the environment and quantify this also. But first part, I will go from left to right, photosynthesis measuring with lift. As Katarina said, this will be a talk about this instrument, so I thought it would be good to give a little bit history of this instrument. A history that started also similar by Roland Pirushka, measuring gas exchange, electron transport rates of the lift and other devices. And at the first stage, you see the name is a little bit different. It's a laser-induced fluorescent transient device. It's also a bit more heavy. It's here about 50 kilograms, and you see it here being carried up with a crane on the roof, or you see it on a tower in Campus Klein Altdorf, the picture here in the middle. showing this laser-induced fluorescent transient device in operation. This laser allowed it to measure from 50 meters distance. Nice, because you can have a reduced distance. Not so nice, because you cannot position it easily at different places. But it learned us the first part, that this lift transient device gives us some functional traits. When I arrived in Jülich and they asked me to do phenotyping, at the same time, actually also more or less a year after, I had the possibility to get my hands on this device, also a lift device, but much smaller. And I put up this picture on the left. That is Spinja Kolber, the person on the left standing there, and behind him, looking over his shoulder, is Barry Osmond. These two persons have been instrumental in bringing this lift instrument to the community, the science community, but also closer to the users community. I must say, Barry Osmond, and you see his surrounding people around it, they're a little bit older than us, but that gives the advantage they have a lot of knowledge, so much knowledge that it goes even further beyond a simple transient. But you see an example of what the lift can do more, and you can look up some publications if you're interested in traits that I will not talk about today. Today, I would like to talk about the main traits of the lift. A lift is active, fast, and it can remote or proximal sensing. And we use it in a way that we have a QA protocol. That means we give blue flashes in a high frequency to the leaf. And these blue flashes, they saturate the photosynthesis process, and you get a saturation, and here you see in red, of chlorophyll fluorescence. After that, we give a relaxing rate of flashes, and this relaxing rate, we will give a relaxation rate of the photosynthesis. This is a new trait that has not been measured so far. So I will talk in this talk about mostly about the maximum rate, the first part of this so-called transient. Here in the next slide, I've defined that a little bit further in the terminologies that are used in this maximum rate of fluorescence. Again, the same transient here displayed against a flashlight number on the x-axis. On the y-axis, you see fluorescence, and in the red line, you see the saturating fluorescence, because we send a lot of high-frequency pulses. What you see also is there the terms that we're using. The FM is a maximum fluorescence, and an FQ is from the 0 to the maximum fluorescence. These two terms, they have been used a lot in photosynthesis. Other devices that use saturating pulses that needed to be clamped on the leaves already defined that this is the photosystem 2 operating efficiency. That with that, you can define the electron transport rate. That was the parameter that I showed in the first slides, directly collecting to photosynthesis gas exchange processes. So this photosystem 2 operating efficiency, we thought, well, if we want to do phenotyping in the field, let us look at this trait. So you'll find back in the next examples a lot of FQ prime, FM prime, and when you see that, then you know that is the photosystem 2 operating efficiency. How can I get these traits out of the field? You saw a black device, about 15 kilos. It's not handheld anymore. So the first person, we can put it somewhere and measure and move plants in front of it. But basically with the device, we can use the whole range of positioning systems available in the community that go from fixed positions to little carriage or big carriage or bridges. And this device, 15 kilo, maybe drones can carry it nowadays, but there it will stop at the drones and the satellites, and we cannot use this device anymore. So to give this theoretical overview what is the community in field phenotyping available, let me show you some examples how we in which devices we integrated the lift, and that's where I summarized. Well, that's what I summarized here below, only in pictures, where you see on the left upper corner. I can see actually if I have a mouse click pointer. Yeah, this. The first example that I gave you, and from that, because the device was so small, we could move it into greenhouses on little carriages, and we can also move it on automatic rails that are hanging in greenhouses. These are in Campus Kanaldorf, or actually most often used, we can put it on kind of bicycles. And here you see different examples. This was in the US in Maricopa, in Brazil, where we didn't bring the bicycle, but they made it for us. This is in Campus Kanaldorf. This is in Mexico. So all kind of different positioning systems, not so different, but the big advantage, they can move. And I will have a video later where you see some movement. This is still all manually pushed. Great to have a PhD student or a postdoc focusing on the trait of interest, FQFM prime. If I want to get in higher throughput, we also meanwhile have a lot of automatic system positions where you see some examples on the pictures. This is what we call the field snake, and this is what we call the field weasel, an engine-powered bridge and an electric-powered bridge that can move the sensor on 1 centimeter accuracy in the field. And on the right upper part is the latest integration of lift, actually last week when I was in Switzerland on one of the robotic systems of Catera. With this, I've given you a very short history of lift, where it came from, what we can measure with it, photosystem to operation efficiency, how we can measure, basically just by mounting it on any kind of carrying or positioning device. And now, in the other half of my talk, I would like to give you some examples of data, what we acquired there with this lift data. And I would like to start with a condition what I call model, and which is just this basic condition where we actually didn't modify the environment. And what we just did, oh, I lost somewhere, this one, is measuring with the lift in high frequency or in high throughput using this rail system in Campus Kanaldorf. You see it here on the lower part. It's a rail system outside of a greenhouse and inside of a greenhouse, as well as this robotic cart where we were driving outside. And you make use of that using this automation, we could collect a lot of data in time and under different light conditions and measuring at the same time photosynthesis. Why do I emphasize this and why I start with this example? Because here the positioning systems really become important. Because we could collect so many photosynthesis data at so many time points, we could actually define this classical Monteith quantitation where we say the total biomass is the sum of light, photosynthesis light interception and photosynthesis conversion, or the last part is actually photosynthetic efficiency. Until now, until these studies, it had been very statically measured. We had 1 measurement, 2 measurements a day of this photosynthesis. And this study of Beat Keller, a PhD student of our group, he acquired many data and allowed to make really a sum of also the photosynthesis measurements, and not only a model. We say this is photosynthesis, and then we see the response to light. And by doing that, he achieved the following. He showed the rates in accumulators over different days, and you see also that the electron transfer rate you see here, you see photosynthesis, you see FQFM, and you see the interesting thing that is not instantaneous measurements in the first figure, and the second figure is the cumulative data. And this cumulative data he used to correlate to biomass. And you could say here on the right side, well, if a student brings home this figure, I wouldn't be happy. Correlations are significant, but poorly. But plant biologists got really excited about it because this was one of the first studies where we could relate a photosynthesis measurement to actual biomass. It's in different conditions and different experiments and relate that. This is something that often doesn't happen because, as you can understand, if you move from photosynthesis to biomass, there are many steps in between. But by having a lot of detailed photosynthesis measurements, Beat could show that photosynthesis contributed significantly to the biomass. That was the one example I wanted to give you as a status quo of doing many measurements over time and the importance of it. These are many measurements over time and they integrate the environment. At the same time, we sometimes do many measurements over time to get a lot of genotypes quantified. And that's the second example where I want to give you an example of a heat stress experiment. Inside the greenhouse, or not really field, you may say, but the greenhouse we used in Italy to really get the heat stress. So it was in summertime, the greenhouses, they are normally cooled. And in this case, we turned off the cooling and we looked at 183 cultivars with the lift. And as you see here, using a kind of bike system together with Christoph Jadmowski and Lars Zimmermann moving over it. And what we got from that is that if you then have a hot day, you can see a decrease in photosynthetic operating efficiency that you see on the FQFM on the y-axis and time during the day on the x-axis. And what you also see that in these box plots, there's a lot of variation. Just to remind you, I was looking here at 183 cultivars, and it's specifically the variation that we were interested in. So now let me pick out some of these genotypes, actually plants that were very responsive to heat stress and plants that were not responsive to heat stress. The blue genotypes in blue, the blue genotypes are the non-responsive one to heat stress, and the red ones are the ones that were very responsive to heat stress. Same trait, measured from the same position of the only different genotypes. And this is information that goes then in GWAS analysis to identify differences between these different genotypes. This was for heat stress as one example of a study that you may not find easily because Lars and Christoph are still working on the publication. A study that you can find back is another example of drought stress. Here we move to Maricopa, a desert in the dry time. So what you see also, the only green part on this picture is the desert. Sorry that I moved. And I moved one moment, sorry. I didn't notice that, yeah. The only green part is the irrigated part in the desert. The irrigated part in the desert keeps it water and is irrigated at sowing. Then they get a crop standing. And what they do then here, you see, I only show you the first year of data. That's the red line. On the y-axis, soil volumetric water content. On the x-axis, the degrees in the days after stopping the irrigation. And you see there a steep drop in soil volumetric water content. And then the question there, as previous in heat, is the question, how do these genotypes respond of during wheat to this drought stress? Are there differences between these ones? And for that, Nicolas was driving with a bike system daily through all this through all these genotypes. And what he got from his data? Well, he again got photosynthetic operating efficiency, here green. He also got the relaxation factors, which I will not go into detail, but both of them show this, if you do it in a percentage stage on relative, after stopping the irrigation, show this typical pattern that you first have an increase in photosynthesis and a decrease over time. This decrease is something we can expect because if plants don't have water at a certain moment, they will die, or many steps before that, they will decrease their photosynthesis. This interesting step, the short term, that they will pump up their photosynthesis a little bit early, is something that the breeders can really use, and maybe even farmers, if this could be really good, quantifiable and comparable, to start the irrigation. So this variation we used further in identifying suitable genotypes that are resistant to drought. And we also wanted to know further, is this a one-time response? One other study showed that, or can we find that in other locations and other breeder populations? So we repeated this experiment, but this time in Mexico. And here I have a short video that you can see how things are sometimes going slowly, but also very simple. And also this experiment, we did it a bit more simple. We choose a tolerant variety to drought, and we choose the variety that is susceptible or responds strongly to drought. And these two varieties, we irrigated them continuously, or we stopped irrigation like at the experiment in Maricopa. And now, well, what you would expect, and remind the colors a little bit, in blue are the irrigated ones. And you see that if they're irrigated, they get a higher yield compared to drought. And you see also that when plants are not irrigated, they drop their yield. And actually, surprisingly, but the tolerant one dropped its yield even more than the susceptible one when it was not irrigated. But there's in both cases a drop. If we then look further into the photosynthesis operating efficiency, this is the domain where we're moving into, can we detect with photosynthesis these drought stresses that result in a yield loss at the end early on. And this is where you see again a time axis on the x-axis and photosynthetic operation efficiency on the y-axis. The symbols are a little bit small, but what you can see is that the in colors that the susceptible variety, where you see here dark to dark orange, actually is decreasing earlier. Here you see it even better in early drought in early drought phase. You can see a drop in the susceptible variety, whereas the tolerant variety maintains its photosynthesis value. So we have an indicator very early on of drought stress that is different between varieties by measuring photosynthetic efficiency. And later in the season, you see basically the same pattern that you see in yield. So if you wait long enough, you could predict your yield as well from this, because you see that the orange symbols, the ones that are exposed to drought, are lower than the irrigated ones. So that was these were the examples of abiotic stress, heat and drought, and photosynthetic efficiency that it can identify it. And we wanted to make our life a little bit more difficult. And that's the next example I would like to give you. And that is in a biotic stress. This is Cercospora leaf spot disease in sugar beet. You can see an example picture on the left. And in extreme cases, what it does, the crop becomes completely brown. And actually, you have a loss and a significant loss of yield in the German Farmer Society. Up to 50% when these leaves get brown. Also, the beets don't produce any sugar anymore. So there the question was in a project that we did from, can we also detect using photosynthetic efficiency the leaf spot disease? And then the best would be as early as possible. So we choose a similar approach. We brought a bike with a lift or a robotic vehicle with a lift to the field. And in this field was a breeder's field of a big sugar beet breeding company. And they scored on a 2-day basis the Cercospora. And we came in and we said, okay, let us look now at the Cercospora scoring of many varieties. And here you see the x-axis, you see the Cercospora scoring. When you high scoring means that they are highly infected by Cercospora, and a low scoring means there are only a few black spots. And on the y-axis, you see again the parameter I've mentioned many times now, photosynthetic operating efficiency. And what I'd like to show you is this decrease. We see that there is a lower operating efficiency if there is a high Cercospora scoring. So we can identify at a certain moment that the Cercospora affects the photosynthesis. The open question, and what I don't really have in this picture, is when can we really detect it? Am I faking a little bit of data now? Is this a stage the plants were all brown and they were really dead, so it's easy to measure photosynthesis, or was this early on? I must say these plants, as you see from the picture here, when we measured them, they were still pretty green, and we were early on, but as the farmers could also identify it. So the question for us, the open question is, can we actually find this trend earlier than as early as possible? That is still an open question. Example of biotic stress. I have 2 more examples in the remaining 5 minutes for you. And that is, we talked about in the course already today as one experimental field that we have been using for dedicated experiments, but also where we allow the students to get the first experience with field phenotyping. And that we do in the breed phase, a facility where we can elevate CO2 in the field. We do that by blowing in CO2, and we control that in the middle of a so-called ring. And for that, we have a positioning system. And you see again, on this positioning system, not surprising, you see the lift hanging, where we can measure over time in the middle of the ring, different crop varieties. In this example, it is winter wheat that we've been growing there for several years, or 3 years to be precisely. And we, as well as other crops like potato and soybean, but I will not show you the data. I would like to show you the data of winter wheat where we measured photosynthetic efficiency. And I would like to combine some data here, because what you see here now on the y-axis is again time of winter wheat that has been exposed to high CO2 over its whole growing period. And what you see here now also is that actually this response to high CO2 and also the photosynthetic response is highly dynamic. If we would have measured one time in summer, in the generative phase, we wouldn't have seen any difference in photosynthetic efficiency. However, in the early phase, you see that the plants on the elevated CO2 actually have a higher photosynthetic efficiency. They can make more use of the CO2 that is available, and they show higher photosynthetic rates. And actually the opposite is happening at the end of the season. The plants that had a high production in the beginning of the season actually had an earlier senescence than the plants that were growing on the normal CO2. So you may think, what is that important now? Well, these dynamics, like the first study that I showed you, we can, and often we do still, look at 1 time point, 2 time points, and we think we can simulate or model the whole process of yield increase. But that's not always the case. And now we have a possibility by this combining a proximal sensing method with positioning systems to go over time and measure these dynamic processes. And we can gain further understanding also in future CO2 conditions, what this will mean with the yield. Right here, I give one example where the yield potential, which means is the actual yield of these crops, how high yield they can get. And you see that the different varieties we selected are these 10 points that you see here, and they varied in their yield. And they were common normal varieties of the breeders. So you see a pretty high ton per hectare production. But what you also see is this positive relation with a relative increase in total dry mass in response to elevated CO2. So plants that actually had a high yield potential also increased their biomass production a lot. And this is interesting, and this is also where we get a mixture of a traditional experiment on the elevated CO2 with an outlook for breeders that plants that have a high potential yield actually are also the plants that under a future climate may show a much higher yield potential than plants that do not have this yield. So, and then we try to understand further the mechanisms behind that with photosynthesis. This is also nice if weather is nice and if the conditions if there's no biotic stress. So the last part is what happens if you combine that. If you look at biotic stress under elevated CO2, and that is here again a lift sensor hanging on a field snake on an elevated CO2 experiment, but in this case it's sugar beets infected and not infected by Cercospora. And we are interested in how does photosynthesis respond to that. In this case, I will not first show you the photosynthesis data, but I'll first show you the yield data. Because this is what is, I think, highly interesting. We had a variety that is, again, susceptible to this disease and a variety that is not so susceptible to this disease. And then you see in this pattern also that we sprayed some plants. The blue patterns are plants that are sprayed with a fungicide, so they shouldn't have any fungicide, and in yellow are the ones that have the disease. And on the right side, the lighter colors is the response to CO2. And what you see here is that for a variety that's susceptible, so that had a high disease, it increases a little bit in response to CO2, and it increases even more if it was exposed to disease. In the variety that was not susceptible, we see a much higher increase in response to CO2. And we can also see, if you compare the plants with disease, that they were indeed very low susceptible, because the root yield here in this variety is higher when nothing happened on the control conditions than the one that's susceptible. So we have here a variety that is responding strongly to elevated CO2 and that is not susceptible to the disease. And there are the questions behind, how do these three plants actually handle, how can they improve their photosynthesis, their yield so much? Can they maintain the photosynthesis over time? And why does this susceptible variety cannot do that? And this is a bit more complex figure, but again, I want to show you over time the seasonal development of FQFM. And I want to show you that we have plants on the ambient and elevated CO2, and the light symbols or the blue symbols are again with fungicides, so they are treated. And the plants, the yellow and orange symbols are the plants that are inoculated. That means they have the disease. And if you first look at the end of the season, you see the plants that had the disease, they also had lower photosynthetic rates. That is in the plants that were susceptible. If you look at the plants that were not susceptible, it really shows also that plants that are not susceptible don't show a difference in photosynthesis at the end of the season. That's kind of confirming, it's directly confirming the different genotypes, and with that also the different phenotypes. So that's nice input that we can measure from a proximal sensing trait, also susceptibility of these plants. In the early season, it gets a bit more complex, because there we see that the plants on the ambient CO2 actually have a higher photosynthesis than the plants on the elevated CO2. And with that, I leave an open question to you, why that may be. The plants that are grown on the higher CO2 actually show lower photosynthesis rates. I have an explanation, but I'm happy that you can actually think about it as well, because otherwise I use too much time to go into this. But here the open question is, why do plants on the elevated CO2 actually reduce their photosynthesis? And that was the last example I wanted to give you in a very short overview, just some examples of the field, pictures and data that are not there, where I hoped I could summarize a little bit how we approach field phenotyping photosynthesis from photosynthesis traits measured with lift, using different different positioning systems in different experiments, and where the environment, in this case drought or water, also plays a role. I'm not sure if I did a good job in doing this, but if I say we do phenotyping of photosynthesis, I say, well, we can always improve our throughput and we can actually measure more accurately and quantify the environment correctly. But luckily enough, or not luckily, interesting enough, last 2 months ago, we actually from external, we got the company, the comments that said phenotyping photosynthesis, yes, we can, which is a very ambitious title. And I'm very proud, I could say, of Beat Keller, because he wrote this paper where they based this conclusion upon, where they said, well, if we measure with the lift a whole population of bean plants, we can actually show the differences in the field. And this study and the other studies of Beat, Nicolas, with the lift in the field, came to this commentary of Samuel Taylor that says, well, actually now the techniques that we have available, and where I showed you the examples of, can allow us to phenotype photosynthesis in the field. Of course, this was not my job alone, and it was thanks to a lot to the IBG Plant Science, Uwe Rasche, and our group of Shoot Dynamics, where a long list of names over the last years where all these experiments were done. And happy to answer any questions or invite you for measurements in the field. As I said, specifically in the PhenoROP experiment coming up this year, but also to look further into other applications that we have, for example, on the solar panels, or a topic that I didn't touch about, what happens if we get a sensor that actually is light enough to be mounted on a drone and give us fluorescence information in the field. I think this will open up a whole new field of field phenotyping photosynthesis. But this is something I hope to talk about in, say, 2 or 3 years. Thank you very much for your attention. Happy to answer any questions.